now honored to introduce Razvan Marinescu, an assistant professor in computer science and engineering at UC Santa Cruz. He specializes in machine learning and its applications in healthcare and biomolecular systems. In addition, he co-founded and is CTO of Tech, a startup focused on innovative drug screening solutions. Today, he'll share his expertise on how synthetic data can enhance the analysis of brain function, address challenges in fMRI, among a list of other topics. Without further ado, please join us in welcoming Dr. Razvan Marinescu.
Thank you so much for inviting me. Like Randall told me about this awesome workshop that you guys have put together, and it's really amazing to see this kind of like community effort towards tackling like a big challenge on like whole brain emulation and how to kind of go about it. It's such a big challenge that kind of like, of course, will take many years, potentially decades, and but it's very important to start thinking about it right now and kind of like lay out a path towards achieving it. I hope what I'm going to talk to you today is about how you can use MRI technology to infer brain structure and function, and I think it's really important for this grand goal of basically doing whole brain simulations or emulations. I'll make it clear in the presentation very soon, but this is basically, as opposed to the previous talks right now, focusing more on individual neurons, like simple organisms like Drosophila or like the worm. This is gonna be about about big brains, like measuring activity at like the whole brain level in humans, and the technology we have to basically measure it. So not just activity, but even structure. I'm gonna start with even more basic things like structure, and then move on to functions. So the plan here, I wanted to talk about MRI simulators first, because they're very important, and why we should be using them, and how we can combine them with current AI methods. Then I was hoping to tell you about how we can infer structure with these types of methods, then about brain function, and then how all of this can basically be used for like the goal of the workshop, which is on whole brain emulation, how we can use all this stuff, all this technology for sort of like calibrating these models we've been talking about. So let's start with the first part. Why MRI simulations and simulators? So I would say the dream of brain magnetic resonance imaging is to basically see inside the brain and map and measure every single neuron, and do all of this non-invasively in vivo, like without having to wait until somebody dies, without opening up their brain and kind of like doing invasive procedures. This would all be done like non-invasively, without harming anyone and any particular human. The dream would be that we want to see individual layers of neurons and how they're basically packed. We want to potentially map individual neurons and see what their structure is for those parameters and find their function. And even not just for whole brain emulation, we want to do it also to characterize any diseases at all, like brain diseases, neurodegenerative diseases like Alzheimer's, for example, like Parkinson's, and so on and so forth, and map biomarkers at all these different scales. We're talking about like here high-level scales, like for example boundaries between structures, substructures, different cortical layers. We'll also be talking about like low-level types of markers, like measuring myelin content, demyelination in some neurons, axonal degeneration. These are all super important for then, like again, instantiating all those models we've been talking about. We want to map even something like tumor macro environments or micro environments, and then maybe the vasculature and perfusion in these tumors, and how that all like affects these types of and our brain function altogether. And we even want to basically get direct to noise-free measures of functional activity. We want to be able to measure function. This is very important. This is what we're going to be used to then like calibrate these emulation basically types of works for doing whole brain emulations. However, this is the dream. Now in practice, like the current technology we have today, like AI technology, MRI, and this kind of like supervised like neural networks, they're not able to do this properly, and it's very, very difficult. For example, some of the types of like state-of-the-art models for doing reconstruction of data from MRI scans currently use neural networks. Like AutoMap, for example, is like a popular type of neural network that can reconstruct from the scanner measurements. They're called K-space data. They can reconstruct the images. But again, we've been talking a lot about the inverse problems and how these pre-inverse problems can be like sometimes ill-posed, and you don't have enough data to reconstruct. So this is again a problem also here, and especially these models have this issue. So, and in particular, they also cannot properly deal with distribution shifts. For example, you have a change in the MRI sequence, or maybe a change in your brain anatomy. Then these models sometimes cannot really generalize to a new MRI sequence, a new anatomy, a different type of noise model, or any changes in, say, patient motion. Like sometimes these patients can move in the scanner, and that can cause problems. And even these types of models cannot allow us to optimize MRI sequences to basically to optimize them for different types of protocols. But that being said, MRI simulators can help us offer really strong physics-based inductive biases. What do I mean by a simulator? So we can actually simulate the physics of the MRI scanner. What is happening in the MRI scanner, we can simulate that in a computer, which I think is awesome. And what happens is that you have in an MRI scanner, you have basically each location in your brain, each voxel in your brain gets magnetized along the main magnetic field, and then you send some pulse sequences and some gradients that change this magnetization of this particular spin at this particular location in the brain. It changes it in a different direction, and then this magnetization relaxes back to the main magnetic field, the Z direction. And this relaxation is what allows us to get contrast, to get data, to basically measure the brain images. Mathematically, this is modeled, basically, it can be modeled very easily and nicely in what's called a Fourier basis. We have the magnetization, for example, the transfer magnetization along the XY plane. That's a steady state, and this basically gets projected into a Fourier basis on these Fourier basis functions. And then we can basically map the evolution of magnetization in terms of these Fourier states, and in terms of this like Q matrix. And then each operation that the MRI scanner does, like RF pulses, gradients, or spin precession, can be modeled as matrix multiplications. Basically, this is our current state of magnetization for example, in the entire brain. We multiply by this matrix, and then we get a new state. And then, for example, if we want to model a radio frequency pulse in an MRI scanner, we multiply by a different matrix instead. And we have also matrices for all these types of operations. So that's how we can basically simulate in a computer the MRI physics. And again, the really nice thing of this is that it gives us very strong inductive biases. We don't have to learn. We don't have to do any deep learning. We don't have to acquire like thousands and tens of thousands of images. We just have it basically there for free. We know the laws of physics, so we can just like simulate them. There's many advantages. We can simulate any MR sequences with this type of approach. We can generate an infinite amount of synthetic data. From any contrast. We can be a T1 contrast, T2, functional MRI, MR angiography, any kind of contrast for any anatomical image as an input. People have a lot of heterogeneity in their brains. Some brains have different shapes, different sizes. We want to measure that. We want to simulate the data for each of these brains. And there's more we can do. Some things are not entirely obvious yet, even to the AI and MRI community. One of the things that we can do is that we can even use these simulators to infer what I call digital twins of the brain. Basically, we can infer structure, function, any of these. Like at each voxel, we can map how much gray matter we have, how much white matter, how much CSF we have, how much CSF meaning it's a cerebrospinal fluid. How many blood vessels, all these kinds of we can do a very detailed mapping of the brain at each location. And finally, we can even optimize with these things. We can even optimize the MRI sequence, so we can get better data. We need better data. We need better measurements, so we can do all this optimization in a simulator. before actually executing on a real scanner. And that's going to be lead to a much faster iterative cycle. The simulators can basically go through thousands of MR sequences and different types of parameters and basically find us the optimal sequence for a particular goal that we want. So I hope I convinced you a bit about MR simulators, but now let's see how in practice, how do we use them to infer brain structure and function? Do we take questions at the end or also during the talk?
We'll take questions at the end.
At the end, okay, all right. So that's fine. So how do we infer structure and function? The first thing I want to mention is that on differentiability. Differentiability is a key property that can help us efficiently optimize simulator inputs. And this is something, again, the community is not fully aware and realizing it yet, but it's starting to basically creep in. Basically, the idea is that we can model the MRI signal here for a given brain. We can model the final signal that is going to be taken by an MRI as a composition of multiple functions. For example, we take the brain, we measure the transverse transmagnetization in the XY plane, and we apply different types of operations like relaxation operators, gradients along the different directions like Y or Z or X, relaxations by a certain time like tau, and even RF pulses of say 90 degrees, 180 degrees, and so on. And then we can basically infer, since this is a composition of multiple functions, or the entire signal is a composition of these multiple functions, we can then, if we make these functions differentiable, that means we can compute a derivative of each function with respect to the inputs. Then we can basically use the deep learning machinery, in particular stochastic gradient descent, to optimize our brain input, our brain image. So basically we can optimize. Given a particular measured signal, we can find what's the optimal brain input that basically when measured by the scanner and gets us a signal, the signal that we simulate matches the signal that's being measured. So we can infer the brain with basically like with what I would call like deep learning machinery and deep learning types of like technology. And then we can further seamlessly integrate these differentiable simulators with other neural networks. For example, we might want to do some post-processing of the output of the simulator. We can do that. We can have a neural network afterwards. We can even have it before. We can do some pre-processing of the input. Maybe we can have a neural network that generates the brain image, or we can have a type of a neural representation like NERF type of model to basically generate our brain scan. So then we can do lots of these types of like integrations with the differentiable simulator in a seamless way and do backpropagation through the simulators basically to even learn these networks altogether. And I should say in particular that if we don't have differentiability, then we basically have to rely on gradient-free methods, such as the simplest algorithm and other things like Nelder-Mead and so on, that are much more inefficient. It's much more inefficient, especially in high-dimensional spaces. So having the gradients is super, super important. So what can we do in practice? So let me show you some works from my lab. So we use these types of simulators, differentiable simulators. We start with the digital twin of the brain. So here we have like a brain that has at each location, we have gray matter, white matter, and CSF, how much percentage of each we have in the voxel. We extract what are called T1, T2, and proto density maps from the brain. And then we pass them through a simulator, this MRI simulator, and to get the measurements from the MRI scanner, which we can use to then reconstruct a simulated T1 weighted image, brain image. Now, in practice, we have a real image that we basically measured from an MR scanner. And we build a loss function here between the real and the simulated one, and we backprop the loss all the way to the input digital twin. So that means we can find what is the digital twin, what is the brain that basically gives us the same signal that we basically measured in our scanner. So it is an inverse problem, but we can basically still account for that and basically fix a lot of these things by having, we can remove the opposeness by having multiple measurements, like maybe a T1 scan, a T2 scan, and so on. This will remove the opposeness, and we can also set some really powerful, interesting priors to basically also help us with the opposeness. And this is something we published just recently at ICML. And let me show you the results. So basically on the left are basically the true ground truth maps for cerebrospinal fluid, gray matter, and white matter. And on the right are basically it shows how initially our model can basically over time basically reconstruct and recover those maps. So we start initially with just average maps. I'll stop it for a sec. We start with average brains, basically like a population average. But then based on the signal, we can, after a few iterations, our estimates converge to the true maps for each of these, for CSF, gray matter, and white matter. So it's a really cool and like novel type of a paradigm, I would say, altogether, because people so far have only been using supervised machine learning to basically supervised deep learning to reconstruct these. This is a method that doesn't require learning and basically has very strong inductive biases, which enable you to actually do much better reconstructions than a supervised deep learning approach. And now to change the gears a bit, we can also talk about optimizing for microstructure, not just macrostructure, but we can basically try to see how can we get to the level of measuring individual neurons, or maybe just groups of neurons, or maybe fiber bundles. This is what is in the realm of what's called diffusion MRI, and you can get these beautiful images of diffusion, what's called diffusion tractography. These are basically white matter connections connecting any arbitrary parts of the brain. And there's millions of this that's being reconstructed. And this is all done in vivo. Like you don't have to basically take someone's brain and slice it and put it under a microscope. This is all done in vivo. And you can do these types of scans and inferences over time. And you can see even how these connectomes change as somebody ages, for example, or somebody develops a disease, and so on and so forth. So it's a really powerful type of a technique. And we've been working on this as well, on building a differentiable diffusion MRI simulator now. And some simulators like this existed, but none of them have been differentiable so far. And we actually built the first differentiable simulator for diffusion MRI, which is based on a type of like model that uses partial differential equations and uses a finite element method that somehow models the diffusion of water molecules in basically like a neuronal mesh, and basically simulates the MR signal that would be measured given a particular type of diffusion and a particular type of mesh. And this again allows us to basically infer the microstructure right now. So much more detailed structure than just like, those kind of like average large scale brain maps. And in particular, we can infer it as arbitrary 3D meshes, which is something that's not been possible so far in the field. People have been fitting just these very simple, what I call diffusion tensors, these like ellipsoids at each voxel in the brain. We can now fit much more complex structures here in each voxel. To give you an example, so this is a type of optimization that our simulator did. So we're starting with a mesh like this, with a cylinder type of mesh like this, and this is the ground truth shape, like it's a bent cylinder, perhaps being part of like a bent axon, for example, here. And over many iterations of 4 passes through the simulator and backwards passes with the backpropagation, we can basically reconstruct a mesh that is very similar to the ground truth mesh, and basically that part of the axon, for example, we can infer it quite reliably, and again completely non-invasively with this. And again, we can also reconstruct not just these types of simple meshes, but even more complex ones, like what I call beaded axons. So beaded axons are actually like axons that have these like bulges, as you see here. And these are actually prevalent, especially in some white matter diseases like multiple sclerosis and so on. Have been shown that the axons are actually not flat cylinders, but they have these like undulations, basically. So we can reconstruct them even with our simulator, even these types of beadings, which is super cool and super interesting. So now I've spoken about the brain structure, which is very important. But how about inferring brain function using functional MRI simulators? Again, this is going to take us closer to basically having functional data on functional activity of the brain, which can help us do later on brain emulations. So let's see how can we measure brain function with functional MRI. So the idea here, some of you might be familiar, some of you might not be, but functional MRI can basically read functional activity of neurons non-invasively using what's called a blood level oxygenation, or what also is abbreviated as a bold contrast. The idea is you have a stimulus at time 0, in a particular location in the brain, say here, and then after those neurons, so basically that means those neurons are firing excessively, and then because of the activity of the neurons, they need more blood. They need the hemodynamic responses that they pumped with new blood, with fresh blood, and that fresh blood basically contains hemoglobin that is I forgot exactly if it was oxygenated or deoxygenated, but anyway, it's the difference in the oxygenation of the hemoglobin is what allows us to basically measure this bold response. So because one of the oxygenated hemoglobin is paramagnetic and the other one is diamagnetic, and this difference in magnetization is what allows us to get this contrast. fMRI uses to basically measure activity live in a live manner. Like it uses a fast sequence called EPI, echoplanar imaging. Normally, an MRI scan takes a very long time, can be 20 minutes, can be an entire hour, but these are very fast sequences that are basically measured very quickly, and then you can get functional activity over time. One slight problem I would say is that the signal is the functional signal is only around 2 or 3% of the total magnitude of the signal, and that means you need to have a very low noise. Otherwise, the actual functional signal will be drowned in the noise. So, like, we have to be very careful how you set up your measurements and so on, and the scanner has to be very well calibrated so you have as little noise as possible. And also, I would say the reconstruction that allows us to get these functional maps. So these maps here show for a particular stimuli. Like, for example, in this case, this subject was doing some hand movements in the scanner, and the scanner was able to reconstruct which areas of the brain are active during that particular activity, like moving their hands. And these are the areas that were being found. So this is what I mean by reconstruction of the functional activity, getting these kinds of maps for a particular task. So this actually does require doing some statistical significance testing, doing some t-tests, computing some p-values, maybe even some more sophisticated tests, not just t-tests, and maybe even accounting for multiple comparisons. But what about simulators now? So the idea is, can we simulate this whole procedure, this whole process with functional MRI, the way we've done it before with structure? And the idea and the answer is yes, we can. Actually, so there have been some simulators that have been built for functional MRI that pretty much work in a similar way to normal MRI. They still simulate the block equation, which models the change in magnetization over time. So here it's basically the change in magnetization M over time is basically given by the effect coming from the main magnetic field B, plus the components over X, Y, and Z direction. That has the relaxed following a T2 decay or a T1 decay. And in particular, again, a transverse relaxation can be measured basically like this. Can also be written with an exponential term like this. So again, the transverse relaxation basically measures what's called the dephasing of the spins along the XY plane, basically. And this transverse relaxation T2 is exactly what is being exploited by the bold signal. So and basically, again, it's basically the bold signal is assumed to be a time-varying function of T2 star in particular. I won't go into detail what star means in this presentation, but think of it as something very close to T2, and this is what is being measured as the bold signal, and the idea is that we can simulate the T2 star by basically taking a stimulus, like some kind of like synthetic stimulus over the whole brain and over time. So this will be a function that's over the space of the entire brain and over time, and we can convolve it with what's called the hemodynamic response function, or the HRF function, to get which is this kind of function. This is the hemodynamic response function here, to basically get the T2 star map, and then we can put in a simulator, and then we can get a signal. To get a bit more intuition, this is the type of this animation kind of shows how such a sequence kind of runs and what this does. So basically, we have a series of spins here that have been changed with like what's called like a 90-degree pulse, and now we're applying a 180-degree pulse, and they're gonna very soon start to enter resonance. You see, here, this is the resonance, is the echo that's being measured as the T2 as a T2 echo, basically, in this case. And then we can run another 180-degree pulse here. And then they're going to echo again. They're going to enter resonance very soon. And here we go. This is the echo, and this is again what's being measured. This is the entire output of the scanner. So, and again, doing another echo, doing another 180-degree pulse, which will lead to another echo. And here we go. So this is the type of physics that is being simulated with these equations. Move the movements of all these spins, and these pulses are what we basically put in the MR scanner as the MR sequence, and then it allows us to basically measure all these kinds of like super interesting phenomena like functional activity and so on, and even structure. There's in terms of the landscape of fMRI simulators, there's actually quite a few by now. That's not the only one that I was showing. There's been a few more recent works. In particular, one of the things people have been trying to do is to basically make them faster. Like some of the types of formalism here is actually the most accurate by far, but they're very computationally intensive to simulate all these equations and integrate them over time. So people have been looking at ways to make them faster, making some assumptions. Maybe you can assume steady state of the magnetic vectors. Things like that. Maybe you can measure right away in a K space or right away in image space, and you don't have to simulate all those things. But then you do lose some accuracy, and you do lose some the ability to simulate certain kinds of effects, like motion of the patient in the scanner, and lots of other things. For example, undersampling of if you do it right away in image space, you cannot even simulate properly the undersampling of K space. That you might need sometimes to make the sequence much faster. And now this is one of my hypotheses that, and it has not been done, but I think I hypothesize that we can also efficiently reconstruct functional activity with such functional MRI simulators. So the idea would be we have the same thing. We have a functional activity map that we kind of initialize in the brain. We apply a hemodynamic response function based on this type of a function which is kind of well known already. We get a bold response. This is synthetic, okay, all of this. We put it in the functional MRI simulator, and we get the simulated K space, the measurements that the scanner would take. Then we have, say, for example, real measurements from an actual scan, and we build a loss function, and we backpropagate this loss all the way to the functional activity. So we can infer this way with differentiability and with the simulator functional activity that basically that was reconstructed from this real measurements here. My hypothesis is that this should work, and this should actually lead to even better type of reconstructions that even better than the ones we've been doing right now. In particular, with this type of work, we can make fewer assumptions. We don't need the voxel independence, and we also don't need to do statistical testing at all. So, and this will lead to, I hypothesize, to much better reconstructions. So actually, it's something that we're currently working on. So if you're interested, do get in touch with me. We can collaborate on something like this. Okay, so in the final part of my talk, I wanted to talk about how do we put it all together? How do we use these types of simulators, MRI technology, functional MRI towards whole brain emulation? How do we? How can this really help? Let's discuss a bit about this. So my hypothesis is that actually bottom-up whole brain emulations will require what I would call high-level calibration. We will need to be able to calibrate these models. So in particular, I would say that some of the most promising whole brain emulation models will emulate activity starting from as low as possible, maybe from the level of individual neurons, ideally. Those would be the most promising if we have the computational resources to do it, and the models, the data to instantiate them. These would be the most promising because they would be able to create what I would call like emergent phenomena, basically, and that would be basically different types of that would basically correspond to different types of cognitive processes that can emerge from these low-level neuronal interactions. Like maybe we could have, like at the medium level, we can have things like we can simulate pattern recognition done by the brain. We can simulate memory formation, motor coordination, attention mapping, sensory processing, basic sound production. Any of these could emerge from low-level interactions between neurons. And maybe at an even higher level, we can basically even simulate things like abstract thinking, creativity, imagination, language, symbolic thought, ethics, and even goal setting and agency, and so on. So all of these could potentially emerge from a bottom-up model if we have enough resources to do it. And here by bottom-up model, I mean, for example, things like spiky neural networks, what are currently being used in quite a few applications. These are types of neural nets that basically simulate entire time series for each neuron of spikes, and basically they have certain types of frameworks and how they can basically accumulate potential, and at some point that potential gets released, and then that sends a spike, and then there's a lot to say, but basically that would be the type of model that I would think would be very promising to try on this. And also going back to the emergent phenomena, this emerging phenomena that will emerge from these large interactions of all these neurons will need to be calibrated with actual measurements of such processes. Like we can actually will need to be calibrated because they might not be initially aligned. We might have some slightly wrong parameters at the low level, and that would lead to something like maybe like a biased type of high level phenomena. So we need to do this calibration somehow. And what I would argue is that functional MRI would be the best choice and the method of choice for calibrating these high-level cognitive processes for multiple reasons. Because first of all, these scans are non-invasive. We can do, which means we can run it in thousands of people and maybe even more, and we can measure this way also heterogeneity in the population. Different people, different brains will have different types of activations, functional activity, and so on. We'll be able to map this heterogeneity in entire populations this way, and measure things in vivo as they do certain tasks, and so on and so forth. And also, as opposed to some other technologies like electroencephalography, EEG, or transcranial magnetoencephalography, and so on. These technologies can basically read deep inside the brain. Like fMRI can read deep inside the brain. Those ones, EEG and so on, usually read more like surface level activity of the neurons. They cannot really get information from deep inside, like what's happening there, which neurons are firing deep in the brain. And also I can say they also can read the entire brain at once. Like this fMRI, we get information at once for the entire brain and over time as well. And I would say that the main disadvantage of fMRI, however, is the low SNR and the spatial resolution. But I do believe this will be mitigated in the future with advances in scanner technology. We'll have better and better SNR and spatial resolution as well. But to show you even a bit like what has been able to be achieved so far already with functional MRI, this is an example of a work which did the visual decoding. So basically people were presented with these kinds of videos here, and they were able to reconstruct from the scanner, like they were sitting in the scanner, they were watching these videos, and people were able to reconstruct their the video from the brain activity. This is how it looks. So they were watching these things live, and this is what the reconstruction just from the brain looks for these particular videos. I think it's already quite promising that we can do this, and you can imagine that in the future this will get even better. We'll get even better reconstructions, more high quality. Imagine this will be maybe in 10 years, 20 years, this will be crystal clear, like just like this, for example. We'll be able to reconstruct at that level. Like this is something that I truly believe can be done in the next 20 years if we invest good effort into MRI scanner technology and also reconstruction methods, most likely based on AI as well, and the types of like physics-based MR simulations models. Yeah, so, and there's another paper, a more recent one from CVPR 2023, where again, people were basically presented with like images, with these images on the top. And these are the reconstructions that they did just from their brain data on, again, on the bottom. And you can detect, you can tell, you can tell the person is looking at some kind of plush toy. This is like, you can tell the person is seeing some kind of landscape with trees. An airplane, you can distinguish an airplane quite clearly from this image, and so on. So it's a person in the snow. So I think it's quite fascinating. We can already do this, and it's just going to get better over the next few years. So let's conclude the bit. So what is coming next? What do we have coming next in this type of direction? So what I truly believe is that MRI and AI technology is advancing at a very rapid pace, and we're going to see more and more types of results, and maybe even groundbreaking results in this, and it could lead to breakthroughs also in disease diagnosis and characterization, not just in whole brain emulation. But in particular, they were playing a crucial role in whole brain emulation, and especially on calibration of these models that we've been talking about so far. That being said, we do need to make more progress in terms of better MRI hardware and also algorithms, AI, and simulators. But yeah, that being said, again, like we might be quite prone to a revolution in brain-computer interfaces and whole brain emulation that basically would allow us to talk about things that are currently perceived as science fiction. Like, for example, we might be able to talk maybe in a few decades or very soon about mind uploading somebody's mind into one of these clouds, and having a digital having a digital avatar of a person, an avatar that mimics a particular person that is alive or that's dead, for example. Imagine having an avatar of your favorite scientist like Einstein and being able to ask them questions every time, oh, what do you think of this, what do you think of that, like a virtual one. And this way, who knows what kind of super interesting applications we could get to with these types of technologies. And we could even talk about artificial consciousness, having these types of uploaded entities in the cloud having some form of consciousness, being aware of their self, having goals, maybe having goals desires, or this kind of things. Why not? I mean, it's a very exciting time. So we might see some of these even in our lifetime. And I'll stop here. Let me know for any questions. I'm happy to take any questions.
I put some questions in, but I think we should just have everyone's questions just come up instead of giving me preference in this case.
Sure. I guess you actually asked the first question. So do you want to read it yourself, or do you want me to narrate it?
Well, I guess I could just pick a question of mine then, because I don't know which one shows up first for you. I have them sorted by popular right now, so I don't know. We'll see what happens. Yeah, I was, so one of my questions is actually just purely something silly technical. Way back in your slides, there was a point where you were mentioning automated configuration, in a sense, of a sequence for your exploration. So I was wondering how you configured the simulated MRI sequences as a space of exploration, if you had any specific tricks or a particular method to that, because I'm interested, because we have a similar automation in mind for the various configurations that we can apply.
And do you mean automation, or do you mean optimizing the fMRI sequence?
It's yeah.
What do you mean by automation, actually?
Yeah, I don't know if you remember which slide it was. It was somewhere way back, but it was something about how in your MRI system, not fMRI, but MRI.
Was it, let's see, optimize the MRI sequence over here, this one?
It might have been. It could be, yeah. It could be that, yeah.
So this is something super interesting. The idea is that right now, so far, these MR sequences have been done by trial and error on an actual scanner. And this is actually a very long and laborious process. People actually go to the scanner, try certain parameters, and see which are the best ones. But taking a scan can take an hour, take more even. So kind of like this process that's been done so far has been super slow. With these types of simulators, we can actually optimize. We can find the optimal sequences for a particular goal, optimal sequences that detect a particular contrast in the image or a particular stimuli. Even maybe you can have a sequence purely optimized for visual stimuli and a different sequence optimized for audio stimuli, or so on. So we can see right now that this is not the case, but I could see that in the future potentially happening. So we can have all types of functional sequences being optimized for different goals. And these simulators can basically optimize the sequences very fast. They can basically try thousands of parameters, different types of sequences, simulate them one by one, and see which one gives you the best signal to noise ratio, the best contrast, or the best reconstruction of the stimuli.
But you do basically have to take it all the way through the process in order to then do the next step and try another optimization step.
Yes. So you have a sequence, you do one four pass to the simulator, and then you go back and you try a different parameter and you do another 4 passes.
Okay. Okay. Great.
Yeah.
Yeah, Bill, do you want to ask someone else, or shall I go and with another one of my questions?
We have a few, so let's move on to another person's question,
Okay.
and we can go back to yours. Yeah. So we have a question from Aditya Rajesh. They ask, what do people misunderstand about what this technology can do already? Where is it being underused, you believe?
That's a very good question. Very good. Let me think. Yes, I think the biggest misunderstanding is that functional MRI can get you very detailed readings of the brain, non-invasively. Let me say why. For example, if you look at the public space so far, in terms of even startups doing brain-computer interface technology and so on, most of them are trying to do brain implants. They think that the most promising way in the future to basically read information from your brain is by basically opening up your skull and basically implanting something in your brain surgically in a surgery. This is like Neuralink, like lots of other startups doing exactly this. I think the biggest misunderstanding is that they're not fully aware of the capabilities that functional MRI can do, and can do this non-invasively without opening up your brain, basically. And in particular, I think that's super important, because then implants, if you make them work, they can be useful nevertheless, especially for people that have disabilities. Imagine somebody who's in a wheelchair, total paralysis. They will be super happy to have an implant in their brain so that they can regain their movements. But the general population will not just have an implant just because they want to basically have the ability to have a brain-computer interface. So as a result of this, I think this will not be super brain implants will not be mass adopted unless we do a completely different technology that's completely non-invasive. So let me say otherwise. I think brain-computer interfaces will not be mass adopted unless we do it non-invasively without implants. So that's why I think is the biggest misunderstanding right now.
Aditya?
Can you hear me?
Yes.
Yes.
Oh yeah, no, thank you for the answer. I kind of also wanted to ask, could you speak to a bit about about costs related to running what you have now?
Running the simulator, cost of running the MRI simulators, you mean, or cost of running a real MRI machine?
For the processing you do,
yeah.
Outside of what the MRI is doing, what do people need to think about? Suppose they want to inculcate it in something they want to do.
I don't understand. Are you talking about a computational cost of running the simulator, or do you mean imaging cost? Okay.
Computational cost, storage costs.
These simulators can be quite computationally intensive. Let me show you. So this work, for example, where we're basically running the simulator and doing this thing basically needs a lot of GPU memory. So basically, to do the backwards propagation through the simulator, you do it on a GPU, but it requires a lot of memory. So you're talking about, for a full image, for a 4MR image, you would need around 500 gigabytes of GPU memory. So that's very large. That you need many GPUs on a single node. So far, what the techniques we've been doing is basically you can kind of break up the image into multiple chunks and do them one chunk at a time. That's one strategy we've been following. Another one is we've been doing these optimizations just on CPU without doing it on GPU, and because this way we can run it, we can store it in RAM, and we have more RAM than GPU memory. And we can still reconstruct. So all these reconstructions you see here, actually these ones were done on CPU, and you can still reconstruct images. Of course, it would take around 20 times slower or even potentially longer, but yeah, but that's totally fine because it's a reconstruction. You're not training a very big AI model. You do reconstructed images instead. So yeah, memory costs are very large. You also need to do iterations. As you see here, like there's multiple iterations, like something like in this case, we did 500 iterations. Like that means 504 passes through the simulator are being done to find the solution to this problem, for example. So you need a fast simulator also. If just one 4 pass takes hours, you're not going to be able to do multiple of them to do an intuitive reconstruction. Yeah. Does that answer?
Oh yeah, that was awesome. Thank you.
Cool, yeah.
We also have a question from Alexander McGlynn. They ask, does the MRI simulator technique only work on living brains, or can it also work on brains that are preserved by chemical fixation?
You can do brains that are preserved by chemical fixation. You cannot do functional MRI on those because there's no functional activity on dead brains. But you can do structural simulations, of course. You can simulate, like, these things here are structural MRI simulations. We can reconstruct the structure, the anatomical high-level structure, or even the microstructure, like we see here, these white matter fibers and stuff. This can be done also on fixated brains. So yeah. We can do these simulations and imaging also on fixated brains.
So Randall, one of your questions references Conrad Cording's points about the inverse problem. Do we want to make sure that everyone's on the same page?
Yeah, sure. So I'll just ask that question then. So having heard Conrad's points about the inverse problem and everything influencing everything else when you're doing things functionally, coming from a totally different field, because you have the benefit of not having been stuck in the neuroscience bubble the whole time, in a sense. Do you know of other domains that struggle with the same or similar problems and the methods that they use to solve that?
Yes. All of medical imaging. So this is, I don't mean neuroscience here. I just mean like a high-level medical imaging. All struggles with ill-posed reconstructions. You have too few measurements for the signal you're trying to reconstruct, and then people have been classically using classical priors like total variation priors, kind of like these like smoothness priors, maybe perhaps in a Fourier basis or a wavelet basis. These are the types of classical techniques that have been used to regularize your ill-posed problem. But what I would say more recently and more promising and new approach is to basically use neural networks as priors. And actually, you can use deep generative networks like GANs, diffusion models, autoencoders, variation autoencoders. You can use them as priors. You can use them to learn a prior distribution of what these brain images or brain functional activity maps can be. And we actually, I didn't include this, but we have a paper from 4 years or 5 years ago, which was doing exactly that. It was able to do a very ill-posed reconstructions with very few measurements, but because he had a very strong prior, like not strong, but a very powerful prior. Those are, to me, the different things. We had a very powerful prior given by a very large generative model like StyleGAN in that case. That was a state-of-the-art model 4 years ago. Because we had a powerful prior, we were able to do super interesting reconstructions with just a few measurements. Like we were able to do super resolution, extreme super resolution. We were able to do in-painting of images. So lots of these are classical applications in computer vision. So like we're very ill-posed reconstructions, but nonetheless they work remarkably well with the powerful priors given by neural nets. So that's what I would say is right now the most promising strategy to do reconstructions when you have a few measurements.
That sounds very promising. Thanks.
Yeah, you're welcome.
Our next question asks, LLMs have definitely accelerated the public conversation of a digital consciousness or mind uploading.
Yeah.
How can fMRI technology sort of bypass that need to do high-resolution data and produce quote-unquote thoughts? Do you know of any techniques on the rise? How can previous talks techniques still help?
Well, I fail to see apart from the LLMs being having the claim to be sentient in some sort of sense, but I don't see the connection of them with fMRI. How? So maybe I'll leave the LLMs aside and just try to focus on fMRI. So the question on fMRI was how can can you repeat the fMRI part again?
Yes. How can
the speaker who asked the question could elaborate? That'll be probably easiest if you want to connect and say it out loud.
Oh, yes, they're anonymous, so I don't know who, but
Okay.
if they're here on the call and they're listening, please elaborate wherever possible. But in the meantime, I can re-ask the fMRI part.
Yes.
How can fMRI technology sort of bypass that need to do high-resolution data and produce quote-unquote thoughts?
We cannot really bypass the high resolution. Like you want high resolution data. Without high resolution data, you cannot get very good reconstructions and basically of your thoughts, of your cognitive processes that you might want to measure. Like you need high resolution data. Without data, you cannot do proper reconstruction. So I just say it's not possible, really. You need both. Of course, you can do you can use these kind of techniques I was saying with like powerful priors, but that's kind of the drawback is that you will end up, if you have too few measurements, you'll end up relying too much on the prior, and then your reconstructions could be off. You could get a reasonable thought, but it might not have been the thought you had in mind when which we're trying to measure. So I would say it's not really, you need high resolution data. You cannot really bypass it. That's in short. That's why we need advances in MRI hardware as well to give us very good, very nice data.
Randall.
Yeah, I just want to follow up with that. I see this, there's a clear connection between what Razvan is saying here and what Conrad was saying, because Conrad is also basically pointing towards, yes, we need to have high resolution. We need to be able to use as much of the physics that we can find as possible. You can make good basis functions that way. That way you have your Rosetta Stone. If you can have strong priors, it's very helpful, but you can't rely on just a short amount of functional recording as a way to predict everything that you ought to find in order to, this is what Conrad was calling the out of domain question. So basically, if you've only trained up your system, if you've only tried to build up your representation of what's going on in that brain based on a short amount or a small amount, not a great amount of coverage in terms of functional recording, then you're not going to be able to solve these out of domain matches. You can't find what would that brain have done in another circumstance, in a sense.
Mm-hmm. Yeah.
We have our next question from Mike DeVerde. They ask, are you familiar with this DARPA non-intrusive tech? Brainstorms.
Yes.
It employs magnetoelectric nanotransducers, MEMS, localized in neural tissue for BCI applications.
Magnetoelectro. So I'm familiar with the DARPA initiative on non-invasive BCI. I'm familiar with it. I'm not familiar with the magnetoelectric transducers that you mentioned. But
can I burst in? Can you hear me?
Yes.
This is Mike DeVerde. I'm an aerospace engineer, just a uploading enthusiast. Okay, so the way it works is it's iron nanoparticles that are in a ceramic coating. You inject them in the vein and guide them up through the brain-blood barrier with magnets. So they're in the brain. They're dispersed. They're near a neuron that fires. The electrical field changes. The magnetic field is changed. That's picked up by a helmet worn on the head. I really love your approach that's non-intrusive, but of course the fMRI, I can't sit there for hours at a time inside one of these things, right? If I can wear a helmet, perhaps you can detect neurons firing as I watch TV, as I talk, and map out thought patterns using a non-invasive technique. So Sorry, that's my synopsis. And it's a DARPA program, so they're trying to basically be able
Yeah.
to communicate with soldiers in the field. I don't care about that part. It's the tech. It sounds wonderful.
I know. So that's sort of like that's a dream in the MRI field as well, to make them portable also. Like right now, these MRIs are big machines. If only we can make it down to a helmet that you could also put on and use it to scan your brain as you walk around, do any kind of activity, or speak to someone, or like have a conversation, any of that, like that would be phenomenal. Yeah, and this way you would hopefully need much, or you would not need any contrast agent or these magnetic particles that go into your brain. And yeah, I agree. I agree with that vision. I really agree with that vision. We have to go there. We really have to go there. Let me stress that. Maybe there's more than just one technology. Exactly. Maybe we could have both. We can have this like transcranial magnetoencephalography. We can have fMRI. We can have, but really we have to go there one way or another. Like so. I really like the vision. So yeah, and people. So I can speak a bit about. I can say a few words about how you would go about making the MRIs portable, because like that's another direction actually the field is trying to get to. So there's this for those of you that might not be too familiar, there's this like recent push on low field MRIs. These are basically MRIs that are not as big as before. These are much smaller MRIs. They can still scan you very, very well, actually, and they don't use as high a magnetic field as the other ones. They actually you can. It turns out you can get good images even with low magnetic fields, and because they have low magnetic fields, they don't need to use superconducting magnets anymore to create. And that means they become way cheaper, and in the order of like even $50,000, you could probably build one of those, or 100. Yeah, which is way a big MRI machine is like $1 million plus, like $1 million plus. So they become way cheaper, and they also become more portable. You can move them around. These are MRI scans. You can actually move them around, like, but on a tabletop, like this kind of like you push this like tabletop around. And there's people even trying to go a step further to even kind of create. I know. I think it's Lawrence Wald from Boston who's trying to even create a helmet, like an MRI helmet, from what I've heard.
Yep.
It's a big challenge. It's like there's a big challenge with like getting it to work with like permanent magnets in a low magnetic field. You might not have a uniform magnetic field, so you might have to do more fancy reconstructions that don't use the fast Fourier transforms. So you can still, it turns out you can still reconstruct even if you don't have a uniform magnetic field, actually. And actually not many people are aware of that. But it does become, the reconstruction becomes a bit more challenging from a computational point of view, from the software. But you can still do it in the software, or even use the neural nets to do it fast. So there's ways to go about it, basically. There's solutions to make it more portable, to make it more portable and cheaper. So yeah, like any of these would be super awesome to get it to
Thank you for your answer, and thank you for this research, because although I really do enjoy, for instance, the fly vision simulations and so forth, my personal interest is in human brain emulation, and you are getting really close. So thank you.
Yeah, thank you so much. Thank you. Yeah.
So our next question is, would precision functional mapping take us closer to digitally reconstructing the human brain? Why or why not?
I think so. I think definitely, because if you have precision functional mapping, that means we can basically we can expose people to all kinds of stimuli, like visual stimuli, auditory stimuli, and we can understand. We can decode the brain states. We can measure the brain states and basically decode them. If you see this pattern in a particular location in the brain, you know what it means. Oh, this is, you're hearing some sound, some high-pitched sound. If you see a different pattern, you know what it means. It's probably you're looking at a particular object or that kind of stuff. So. And then we can go a step further. We can map, like, if you see a different pattern, you can tell that's a language, kind of like it maps to a certain language function and so on. So, and this way we can basically decode very accurately the brain states. And then we can, well, first of all, we can map the heterogeneity in the population of the brain states. Different people will most likely have different patterns for the same brain state, like for the same stimuli, that pattern might be shown in different way because their brains are different. And once we map this heterogeneity in the entire population, then we can basically use that to calibrate our whole brain emulations. Maybe we want an emulation of a female brain, like or like something like like a young child who's like 3 years old and is watching a movie, that kind of stuff. So like each or so on, or maybe like you can emulate the brain of somebody with dementia who's 67 years old and has neurodegeneration and has a memory loss. So each of these whole brain emulations for all these different people would be completely different. We want to be able to emulate each of these, I would argue, because then we can basically I think it takes us basically to the direction of mind uploading. We can create a digital consciousness of their mind in digital form, and potentially have again digital avatars of them, of how and this might be again, this digital avatars is something that I see as a big application of this whole brain emulation, I would say, in a sense, because people would like to interact with somebody, like a digital avatar of a particular person, maybe one of their family members who passed away, or that kind of stuff. They might, sometimes it might help them emotionally a lot, or all other kinds of applications I could see. So I think we do want all of these.
So Aditya asks, a bit of an aside, is kernel very good at this cheap portable scanning? Does anybody have experience with it?
What do you mean by kernel? What do you mean by kernel?
Oh, it's a company I know of, and I recently found out the founder also was interested in brain emulation, and so it's a helmet. It's a helmet and it scans. So I guess if I was just curious if you've heard of it.
I haven't. I'm sorry.
Okay, cool.
Yeah, I can jump in there.
Yeah.
So Kernel has indeed made this helmet. It's based on FNIRS, so near infrared. And this means that it is trying to do, to a degree, what MRI is doing. In other words, it's picking up on blood oxygenation, but it only does so up to a certain depth. It can't do what MRI does going all the way through the brain. And the resolution isn't as good either, because they only have a certain number of sensors. So I wouldn't say that it's quite fMRI equivalent or MRI equivalent, but it's a lower resolution version that you could use in a variety of, say, psychophysical settings, or maybe in a medical office or something like that. That was the idea anyway. And I don't know if they've gone anywhere else with it so far, because I was one of the founding team at Kernel way back in 2015, but then I've been out of it since 2017. So I don't know where they've gone with it since then.
Okay. Oh, awesome. Thank you.
We're about to wrap up, but we have a question from Alexander McGlynn. They ask, how fine would the resolution need to be for a successful reconstruction of brains? Is it plausible for MRI to reach nanometer resolutions?
Let me say it right now. So what's the case right now is that one single voxel of fMRI is around 1 millimeter cubed. In 1 millimeter cubed, you have around 5,000 to 8,000 neurons in there that are averaged out. So basically, you average out the activity of 5,000 neurons in one single voxel. So if your question is about how can we get a full reconstruction, more in the sense of being able to map activities of individual neurons, where we're still quite far on that, unfortunately. We need to go way, some orders of magnitude, more in resolution. But that being said, at the same time, I don't think we need to go all the way there to get a good, a successful reconstruction to basically decode your brain states. We don't need to actually measure individual neurons. I hypothesize there's a lot of redundancy and how in the neurons, like for example, if for any particular cognitive process, you'll have lots of neurons firing in a distributed fashion. So you don't need to actually, only if you measure enough of them from that collection, then you'll be able to basically decode that brain state. Yeah. So can they go all the way to nanometers? Not anytime soon. But it's not impossible. That being said, like it's not. I don't discount that in the future as becoming possible. Right now, it's not anytime soon that will not be the case. We're gonna go to nanometers. No. Yeah.
Yeah, thank you. And thank you everyone for your questions. And thank you, Professor Marinescu, for your, presentation and for fielding all of our questions. I know that we ran quite long, and so
thank you so much. Thank you. Yeah, it was awesome. Yeah, great to see all of you here. Like, excited about this. Yeah.